Hello everyone, my name is Rika MIZUI. My presentation theme is genetic engineering. Do you know genetic engineering? I expected a lot of people know this technology. I conducted a questionnaire survey to 30 people whose age group is teens to 50s. Amazingly, 27 people out of 30 answered they don't know this technology. Genetic engineering is the center of attention, and this technology is advancing day by day. This will be used commonly in the near future. However, the number of people who knows much about this technology is still small. In this presentation, I want to consider how genetic engineering will change in the next 20 years and also want to make a chance for the audience to consider their ideas of genetic engineering. First, I want to talk about what is genetic engineering. I'll show you a video lecture, What is a Gene? This video was made by the organization stated clearly and explains the function of a gene.
Stated Clearly presents What exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? You might have heard that there's a blue-eyed gene, a freckle gene, possibly even an anger gene, but single genes don't literally make things like eyeballs or freckles or temper tantrums. Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1 million. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Hemoglobin, for example, is a protein structure found in red blood cells. Its unique shape and size allow it to capture oxygen molecules when blood flows near the lungs, and then release them later when blood flows near oxygen-starved tissues. Pepsin is a digestive protein. Its unique shape allows it to break down food inside your stomach so it can be absorbed by the body. Keratin is a structural protein. Its unique shape and size allow it to link together with other keratin proteins to form hard structures like fingernails, claws, and beaks. Different creatures have different genes, which is ultimately why their bodies look and function differently. But one of the many reasons scientists believe all life on Earth is related is that the basic DNA code, the language of A's, C's, T's, and G's, is pretty much the same for all living things. Many creatures even share some of the same genes. You might not be too surprised to learn that humans and chimps, which are closely related, share 96% of their genetic code. But what would you think a lowly fruit fly has in common with a beautiful swimsuit model? Surprisingly, about half of its genes. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein. Engineers have mixed and matched the genes of different organisms to produce many new creatures, including corn that is toxic to insects but supposedly safe for human consumption, tomatoes that last up to twice as long in the grocery store before going bad, and a new form of bacteria that produce the human protein insulin, which we then collect from these bacteria and give to people with diabetes who need extra insulin to survive. So just to sum things up a bit, what exactly is a gene? A gene is a special stretch of DNA, not the entire strand of DNA, just a segment that codes for something. Each gene is like a unique recipe, which usually tells a cell how to make a protein or a group of proteins. Different creatures have different genes, but all genes are written in the same basic DNA language of A's, C's, T's, and G's. I'm John Perry, and that's genes stated clearly.
This video shows each gene is like a unique recipe which usually tells a cell how to make a protein or protein group. So the most important key of genetic engineering is gene. The next topic is gene therapy. Do you know what is a gene therapy? Gene therapy is one of the technologies to cure disease. The way of gene therapy has 3 steps. First is to take stem cells from patients. Second is this step. To prepare the vector that encapsulates therapeutic gene and deliver them into the stem cells. Once the vector reaches inside of the cell, the DNA becomes expressed automatically by the cell function, and the DNA makes therapeutic proteins. Final step is to get an injection to take in therapeutic protein. So all patients have to do is to have their stem cells extracted and then get an injection. So these steps are very easy for patients. The present condition and subject of gene therapy shows the following data. 80 patients out of 20 who got gene therapy for X-linked combined immunodeficiency disease got positive effects, and 5 people got leukemia. On the other hand, all patients who suffered from adenosine deaminase deficiency got positive effects after gene therapy. The DNA that is involved in our cell is different depending on the kind of disease the patients have. That's why whether or not the result of gene therapy is effective is changed by the kind of disease. Therefore, the way to prevent patients from getting other diseases is not clear. However, the study to research the function of each part of gene is advancing, and it will be clear what gene is effective for a particular disease in the near future. Maybe 20 years later, people will be able to receive gene therapy to treat cancer or other diseases naturally, as if they get an injection to treat a cold now. Third topic is prenatal diagnosis. There are 2 common ways: ultrasound examination and amniotic diagnosis. As the figure shows, we can see the face of baby or the shape of spinal column very clearly by ultrasound examination now. If the shape is not normal, the baby has possibility to have the disease of spinal column. If doctors find some deformity on ultrasound examination, they recommend more detailed amniotic diagnosis. This examination is to check a gene and DNA by using extracted amniotic fluid. According to the result of my questionnaire, 4 people out of 30 answered they understand the way of prenatal diagnosis. In addition, the number of people who answered yes and whose age group was 20s is only 1. The young generation does not know prenatal diagnosis well. 20 years later, the technology which people design their own baby as they want will be established. When teens and 20s become parents after 20 years later, the accuracy of these technologies should be higher undoubtedly. The young generation should understand prenatal diagnosis and have their own ideas. This is my conclusion. In the next 20 years, medical technology and genetic engineering will be related closely. When people go to a hospital, they will be able to select genetic engineering easily as the one way of medical treatment. This is very effective and dreamful technology. However, the spread of this may change their point of view and people may be able to control their lifespan or their child. It will be more important to understand genetic engineering and to have their opinion about life than now. This is reference. Thank you for listening.